Good evening and welcome to the program this evening. I'm Susan L., Vice President and Executive Director of the Foundation for Barnes-Jewish Hospital, and it is my absolute pleasure to welcome you to our final Leading Edge program of the year, this year, 2021. It's a special year for the Foundation as we celebrate 25 years of stewarding your generosity and the generosity of many donors as their gifts help save lives, enrich lives, and transform healthcare. Over these last 2.5 decades, BJC Healthcare has seen remarkable growth. Your philanthropy has inspired research, advanced caregiver educational opportunities, and provided innovative patient care, making possible a healthier community. Your gifts to the Foundation support all areas of care at Barnes-Jewish Hospital on the Washington University Medical Center campus, and nearly every department at the medical school. Now, tonight's Leading Edge Medicine program highlights the Foundation's partnership with the Division of Neurology and the innovative work of Dr. Randall Bateman. Dr. Bateman's research into new technologies, therapies, and drug treatments for Alzheimer's disease is made possible in part by our incredible donors who make a difference every day. Like you, I'm looking forward to his presentation. Randy's internationally recognized for his major contributions to a greater understanding of Alzheimer's and related dementias. He's a recipient of the Potamkin Prize, which is referred to as the Nobel Prize of Alzheimer's disease research. And he is a board member of the National Academy of Medicine. At the forefront of global research in Alzheimer's disease, Dr. Bateman has pioneered a blood test technology that allows for early detection and the monitoring of the production and clearance of critical proteins in the brain. And I'm sure that's what you've all heard about and why you're here tonight. So before we begin, I would like to acknowledge the members of the Foundation's Board of Directors joining us tonight. Thank you all for all that you do on behalf of the Foundation and our hospital and our community. And thank you to our Foundation team and the Division of Neurology and Dr. Bateman's team for the planning and preparation and making this evening possible. Now, we would like to make this as interactive as possible, and so throughout the evening, please be sure to enter your questions and your comments in the chat box to the right of your screen. After Dr. Bateman's presentation, we've arranged for time for Q&A, question and answer, and then after that, our chairman of the Foundation Board, Dick Miles, will conclude the program. And now, it is a great honor for me to introduce Dr. Randy Bateman.
Thank you, and I'd like to especially thank the Foundation for the Barnes-Jewish Hospital for organizing and hosting this presentation, and many of you are watching virtually, so thank you for dialing in and listening to what we have to say about some of the latest advances in Alzheimer's disease. I'm going to cover a fair amount of ground. I'm going to start with some of the basics of Alzheimer's disease, and then I'm going to move to what is the latest in scientific advancements that have enabled us in the field of Alzheimer's to be able to detect some of the changes that occur in Alzheimer's and make even diagnoses with a simple blood test. As part of this, I'm going to describe how research at Washington University and with the newly formed Stable Isotope Labeling Quantitation Center have enabled our ability to identify new biomarkers and new factors that allow us to track the progression and the changes of Alzheimer's disease in real time, and what a transformational change this is for us in a new era of being able to diagnose people in a straightforward fashion. There's a large number of research efforts that contribute to these kinds of studies. I'm showing my research support here from the National Institutes of Health, philanthropic supporters and foundations, as well as pharmaceutical companies that chip in to help in terms of trials that are being run where we're trying to treat and one day prevent Alzheimer's disease. Importantly, a company that David Holtzman and I co-founded about 14 years ago, C2N Diagnostics, now has a clinical test based on some of the technology that I'll describe and that we reported in the field. So Alzheimer's disease is a modern challenge, And it's something that faces us individually in our families, but also faces us as a society just due to the sheer numbers of the people affected, the impact it has on individual patients and their families, and our society in terms of cost and burden. Currently, we don't have a highly effective treatment or a cure, and Alzheimer's, if this doesn't change, is universally fatal. And so it's a stark picture that many people are very concerned about, and it's one of the reasons it's been listed as the most feared disease in people over the age of 65. There are currently more than 30 million people around the world who have this disease. It is the 6th leading cause of death, costs more than $200 billion a year in the US. And those facts are the reason that many of us have taken up in the fight against Alzheimer's. And I'm going to tell you about the latest science that gives us reason for hope and how this is going to change in the future and hopefully the near future. This is a photograph of 2 hemisections or half sections of a human brain. And on the right side, you'll see that that is a normal looking brain, full, without much space, and larger than the brain on the left, which is a brain of a person who's died from Alzheimer's disease. And on the left, you see it's shrunken and it's atrophied. And so this is the organ of Alzheimer's disease. It's our brain, and Alzheimer's disease affects our brain in a way that causes it to get damaged, shrink, and lose its function. When you zoom in on the brain tissue with a microscope, you can see the pathology or the damage that Alzheimer's does on a microscopic scale. On the left side, this dark, cloudy looking structure is what we call amyloid plaques. And on the right side, you can see a dark flame-shaped structure near one of the neurons, actually inside the neuron. We call those tau tangles. These are the 2 pathologies of Alzheimer's disease that are diagnostic and characteristic of the disease. It's how we define Alzheimer's. And over 100 years ago, when Dr. Lois Alzheimer's described these pathologies, it started the modern scientific research into this disease that has now become so common. Shown in this video, you can see the changes of these amyloid plaques in people while they're alive. The growing colors from the purples to the blues, greens, yellows, and reds are accumulating in years over time. 10 years before, there's substantial amounts of amyloid plaques throughout the brain, and you'll see the counter tick up towards 0, which is the year that the first symptoms are noticed. And notice that by the time symptoms are noticed, the brain is already heavily laden with amyloid plaques, and the disease continues to progress. So this was some of the first evidence that we had that the plaques start many years before the first symptoms begin. They continue to grow over time, and then they reach a maximal amount by the time symptoms begin. Shown here is a brief history of Alzheimer's disease development for diagnostic tests. As I mentioned, it was over 100 years ago in 1906 when Dr. Alzheimer's described the first cases. All the way through until recently, the past decade, the standard diagnosis could only be made by brain biopsy or at autopsy. But then in the 80s and 90s, proteins were discovered, and genes that cause the risk and sometimes cause Alzheimer's disease were discovered that led us to focus on 2 of these proteins that make up these pathologies, amyloid beta in the plaques and tau in the tangles. Tracers were developed, and you just saw one of those tracers. It's what we call a PIB-PET tracer. It tracks the amyloid plaques as they change over time. And those plaques we can track in real time in living people. So we don't need autopsies or brain biopsies before we know someone has Alzheimer's. We can now use these kinds of biomarkers to detect when the Alzheimer's disease process starts. Cerebrospinal fluid markers, which we obtain by what's called a lumbar puncture or a spinal tap, also has these proteins in it, and we can use that fluid to identify it. And what's especially exciting is just over the past few years, we have developed ways of detecting Alzheimer's in a blood sample, so that spinal taps and PET scans may not be required in the very near future. Why are blood samples so important? Why is a blood test really a huge advantage? Shown on this slide, I've listed some of the many advantages of a blood test over a spinal tap or a PET scan. But perhaps the greatest impact will be what we're doing in the clinic and what we're doing for clinical trials. So our ability to identify people who are at risk of Alzheimer's disease or who have Alzheimer's disease with a simple blood test means many more people could be identified. We can have much larger trials. We can do them more quickly and for less cost. And that one particular point, more quickly, is perhaps the most important thing we need now. We need to move more quickly towards effective treatments. Why haven't we had blood tests over the past 10 to 20 years when we've had PET scans or cerebrospinal fluid tests? It's because it's not easy. And so the field has tried for the past 2 to 3 decades to develop these kind of blood tests. Shown here is a summary of all the different kinds of studies that have been done on blood amyloid beta. But because the technology needed and the precision needed to identify this needed to be so precise, it wasn't until the past few years that we had the ability to measure this. And so what I'm going to describe now is how, at our center, how we developed the approach of looking at amyloid beta in the blood, how we discovered what the changes meant, and then how they're being used, and then I'll move on to other newer, exciting biomarkers. So shown here is a diagram of a person sitting in a bed, receiving an infusion into one arm, blood samples taken out, and cerebrospinal fluid samples taken out before they're processed and measured by a mass spectrometer. This is not a simple test. This is a highly intense research study where we sample cerebrospinal fluid once an hour over 36 to 48 hours. We use stable isotope labeled amino acids infused into the bloodstream, labeling the proteins that that person is making. And then we analyze it with advanced machines called mass spectrometers to measure which proteins are labeled. But that's how discoveries are made. Sometimes it takes hard work. Shown here in this diagram is an analogy of how this works. If amyloid beta and tau are these cars in blue and yellow, and they're at a traffic stoplight, normally these cars and the proteins transport themselves and clear away, but sometimes things get backed up. And when they get backed up, it can cause a jam in the protein processing. And as those proteins get jammed up and build up over time, that can lead to the pathologies that we saw under the microscope, the amyloid plaques and the tau tangles. And so one way to track these is to use this stable isotope labeling kinetic study that I just talked about, where we track how fast the proteins are moving. How do they come up to the intersection? How are they cleared away? How are they related to each other? And by tracking the speed of the proteins and their turnover rates and how they're produced and how they're made and how they're cleared away, that allows us to understand what's happening in the brain of people who have Alzheimer's disease or don't. And so a lot of our initial work started with a very simple question, which is how do we figure out what's the problem of Alzheimer's? Why do these plaques form? Do people make too much of this amyloid beta? Do they have problems clearing it away? And now what I'm showing you is not an analogy, but the actual data from people who have Alzheimer's disease or plaques of Alzheimer's disease caused by a mutation in a gene. And what these curves show you in real time over hours is the model of how this changes over time. So over just hours, the brain produces amyloid beta, it clears it away, and in people who have amyloid plaques, some of it gets stuck in the brain. And that's what you can see on this image. The red, the growing red plaque is not drawn to scale, but it's quantitative. That's the plaques that are growing over time. So a process that builds up over 20 to 30 years, we can monitor hour by hour in living people using this kind of technique. That allows us to make measures of what's changing and how much is changing, and that enables us to then make better estimates of how we can try to treat the disease. How can we stop plaques from forming? How can we lower the production of amyloid beta, or how can we clear away these amyloid plaques? These studies led us to other approaches, like in this study where we sampled blood directly from the brain. So in patients who were having a medical procedure where the catheter was placed in the brain, we could obtain blood samples and compare it to the blood going into the brain versus the blood coming out. And what we found surprised us in that a lot of the amyloid beta that the brain makes is pumped into the bloodstream. And it ends up in the blood. And you can see this on the right panel. The leg, the peripheral shown there, doesn't produce any amyloid beta or put it in the bloodstream. But to the right of that, the central, that's the brain, the central nervous system, actively pumps amyloid beta into the blood. And because this surprises, because we thought the blood-brain barrier was a good barrier, it would stop amyloid beta from leaking out from the brain, but there's a process, probably active, that moves the amyloid beta to the blood. And this led us to the next question, which is, well, if amyloid beta is being moved to the blood, and maybe about 50% is going into the blood, why is that? And why can't we detect the changes in the blood that people have been looking at for so long? So we did this kinetic labeling study in people with Alzheimer's disease, and in this case it's late onset sporadic Alzheimer's disease, and tried to answer the question of, do the kinetic changes that we saw in the cerebrospinal fluid, are those also seen in the blood? And what you see in the red line is that yes, the changes that we saw in the brain are also seen in the blood. So the people in the red line, the red triangles, those people have amyloid plaques. The people in the blue circles don't have amyloid plaques. Normal is a flat line. The 42 and 40 should have the same kinetic rate. And you can see the red is clearly abnormal. It's slanted and going down. And so that led us to then ponder further, why don't we see changes in concentration in the blood that so many people had measured? And it turns out when we looked at it, there were changes in concentrations in the blood of the amyloid beta 42 to 40 ratio. And that's shown here. So on the left side, this is a much larger study where we collaborated with people from around the world in the US, Europe, and Australia, and other places. They had large cohorts of patients that they had studied carefully over time, and they sent us their samples and we measured them, sent them the results back, and then we were all unblinded as to what the results are. And what you can see is there's a big difference between the red and the blue, the positive amyloid plaques by amyloid PET versus the negative people, the people who don't have amyloid plaques. And so this indicated to us that in fact the blood amyloid beta 42 to 40 ratio is a very good marker of amyloid plaques in the brain, and that we could use this to help identify with a single blood sample who has amyloid plaques in the brain. This is a quantitative measure. We use this kind of analysis. It's called a receiver operating characteristics, area under the curve. What's important about this is a perfect test, one that's 100% accurate all the time, will look like a square. It will go straight up, straight over. And what you see in these lines, the red lines and the yellow lines, is that it's almost a square, but it's rounded off at the edge. And that's because the test is not perfect. Sometimes it says people are positive. When we look at by PET scan, they're negative. And sometimes the test says people are negative. We look by PET scans and they're positive. And so the next question is, well, it's 85, 87, 90% accurate. That's pretty good. That's better than what we have in the clinic today, which is we don't have a test that we can use. And so that makes it accessible, but it's not 100%. And so then the question is, well, why isn't it 100%? And so Suzanne Schindler at our center led this study, and she took the samples from the Knight Alzheimer's Disease Research Center, and we analyzed them in the lab. And what she did was really interesting. She took samples from people who were then followed over time and had multiple samples over time, blood samples, but also had PET scans over time. And then asked the question, when do these change? And it turns out that the blood amyloid beta 42 to 40 ratio changes even earlier than the PET scan, and is one of the reasons why there's a discrepancy between the blood test and the PET scans. And so on the right panel, the right graph, people at year 0 had a blood test done. And some of those people were negative with a PET scan and they stayed negative. That's the black line. And some of those people were negative and they became positive. That's the red line. And so if you have an abnormal blood test at year 0, more than 50% of people have a convert to a positive PET scan within about 5 years. So having an abnormal blood test is a risk net factor now of detecting plaques by PET scan. It's probably just more sensitive. It's picking it up before the PET scan can detect it. So it's an earlier test, and we estimate somewhere around 5 years earlier. So since this time, there's been a ton of development. The field has exploded with blood-based biomarkers, and it's a wonderful thing. There's all kinds of biomarkers being developed because they, as I described, they're so important for us in the field. And one of the things is that, well, not all tests are equal, and so we sometimes will compare this, and this is a recent study just from a few weeks ago where a group in Sweden compared different tests on the right or in the US on the left, and what they found is that the tests that use this high precision mass spectrometry do really well. And in fact, our test did the best. I'm very proud of this. When we found out, we didn't know this, they did the analysis and told us this. So when the lab found out, we celebrated. We had a big round of applause and a big cheer and everything. And so, you know, of course we're not stopping there, and there's much more to do, but it's great that people here at Washington University and in St. Louis can be one of the world's best tests, for example, in Alzheimer's. So we're so proud of everybody in the center. I'm going to switch gears because I've been talking about amyloid plaques, amyloid beta. And you'll remember that at the beginning I mentioned there are two pathologies of Alzheimer's, amyloid plaques, but there's tau tangles. And so what about tau tangles? It turns out tangles are incredibly important for Alzheimer's disease. Amyloid plaques probably start it, they probably kick it off. But the tangles seem to be more of the effector part of the disease. And so we really need to understand better how to track that particular pathology. I'm going to go back a slide and just point out a few of the team members here. So on the top left, Nico Barthelemy is an assistant professor at Washington University in our group and SILQ Center faculty member. Kanta Hori on the right comes from Japan, and he's also now in the SILQ Center. And Eric Madej at the bottom left, I showed you the brain scans, works in the dominantly inherited Alzheimer's network and leads several efforts, including a primary prevention trial. So we all got together and we thought about, well, what can we learn about tau? And are there special things that can tell us about this? And so in this diagram, you'll see that it's complicated, but there's really two parts to this. There's the left half, which is in blue, and the right half, which is in kind of a red or a pink. And on the left half, those are all the pieces that we've known about for 30 years. And we've been studying these, and we're learning lots still about those pieces. We're learning lots about the left half of the protein. But it turns out the right half of the protein that is in the tangles, it's what makes up a tangle, we haven't really studied that in the fluids, in the CSF, in the blood as much. And so this is one of the latest breakthroughs, that we can now study the right half of this protein that ends up stuck in the brain. We can actually study it in the fluids. And this is really quite important. So some of the initial work that Nico led was in identifying all the different shapes and forms of tau. So amyloid beta is complex. There's dozens of forms of it that we study. Tau has hundreds of forms that we study. It's incredibly complex. And what Nico and others identified was that there's characteristic changes, signatures, little markers on the protein, we call phosphorylations as one example, that are highly specific for Alzheimer's disease. And they can be found in the brain, and they can be found in the CSF, and they can be found in the blood. And so you see here in subpanel H where it says plasma, that's blood plasma. These markers in the blood can be highly precise for Alzheimer's pathology. And what we learned was that these markers actually are markers of amyloid plaques first. Even though they're made of tau, it's a tau reaction to amyloid plaques. And they go up very early, but there are other ones that come up later. And so now I'm going to talk about the right half of that protein, this microtubule binding region. It's the right half that gets stuck in the brain and makes up the tangles. Shown in all these graphs is work that Kanta led, where he showed that the parts that deposit in the tangles are really important, and they all go up with disease, but many of them plateau out. But there's one particular region that seems to keep going up, and the more tangles you have, the more it goes up. And the far panel on the right, you see a line, you see that line that goes through those data points. That indicates that there's a very strong relationship between the amount of tangles in the brain and this particular biomarker. So we're discovering specific markers that tell us something about what's happening in the brain, and we can now measure that in the fluids. So this is a map. This is a time scale, a timeline of Alzheimer's disease. And you'll see, going back in years, 0 is the first symptom onset when people first begin having trouble remembering things. If you go back 10, 15, 20, 25 to 30 years before the first symptoms, there are some subtle changes in some of these fluid biomarkers in CSF and blood. First, the amyloid beta goes up, then the phosphorylated tau 217 increases, and phospho-tau 181, other phospho-tau species I hadn't mentioned, 205, other proteins like neurofilament light chain, total tau, and finally the tangles appear right at around symptom onset that they start growing and spreading. And that continues, and you'll see symptom onset 0, 5, 10 years after. So if you take the whole disease process, that's a 40-year time span of a disease process. That's a long time. But it's a window of opportunity. There's a time period before people get sick of 30 years where theoretically we could identify people at risk, maybe stage where they're at in the process, how much time until the symptoms begin or tangles form. And there's a period afterwards now where we can track all these processes after symptom onset. And we can use these biomarkers to identify what stage people are in, and importantly, what happens to the brain when we try to treat it, when we try to target the amyloid plaques or target the tangles. What happens then? So I'm going to switch to a third protein, which is not specific for Alzheimer's. This is what we call a neurodegeneration protein. And it turns out that most things that damage your brain or your nerves cause this marker to go up. So if you fall and bang your head or in a serious accident, if you have a disease like a stroke or multiple sclerosis or a neurodegenerative disease like Alzheimer's disease or Parkinson's, this marker can go up. This is called neurofilament light chain. It's one of the structural proteins. It's like scaffolding in the neurons. And as I said, it's not specific, but that's what makes it useful and powerful, is that it's not dependent on amyloid plaques or tau tangles, and it's a marker of how much damage is occurring. So we use this to track how that's done. Now, the focus in the field right now is on these three kinds of measures, amyloid, tau, and neurodegeneration, but there's many more. Inflammation, how the immune system reacts to the brain. Vascular markers, how do the blood vessels interact with the disease process, and what do they have to do with it? And so really what we need to do is create a good, complete map of everything and track how it's being used. I mentioned that perhaps the greatest thing that we could save in developing these kinds of measures is time. The time it takes us to run a clinical trial, even a short Alzheimer's clinical trial, is 3 to 5 years. That's a long time to test a single treatment. The time it takes us to do a prevention trial is 7 to 10 years. So we get to the end of the trial, and then if that drug doesn't work at that dose in that patient population, we then have to go back to the drawing board. We don't have the time to wait 3, 5, 7, or 10 years to get answers. We have to be able to move more quickly. We have to decrease our cycle time, move more quickly, and get more shots on goal. And so this is the power of something like a blood test. Is one of the things that slows us down is the time it takes to enroll people into the study. So if we could identify people much more quickly and be more representative of the general population and enroll many more people, these trials could be accelerated and complete much faster. And so in this chart you can see on the left, sure, one can save millions of dollars by implementing blood tests instead of more invasive or expensive tests. But the real advantage is the chart on the right. The time that's saved by using blood tests can be cut dramatically. And if we can cut our time by 20%, 30%, or even 50%, imagine what we can do for trials. So I'm going to end with a description of a newly formed center at Washington University. And it's supported and in partnership with fundraising from the Barnes-Jewish Hospital Foundation. And it's called the Stable Isotope Labeling Quantitation Center, which is a mouthful, so we came up with a clever acronym called SILQ Center, so we say SILQ Center. I want to draw attention to this, to the logo of the SILQ Center, because it's actually meant to be scientifically indicative of what the center is about. You might note that in the center of it, there's an atomic nucleus with orbits, maybe of electrons. So we study individual atoms in the proteins of Alzheimer's disease. We actually weigh individual subatomic nuclei. You'll also note that the N in center looks almost like a magnet, and we use powerful magnets inside these mass spectrometers, electromagnets, to control the ionized proteins that are flying through a vacuum like space. And finally, the blue ring of fire is meant to represent that before those proteins get in there, we charge these things with 4,000 volts of electricity. We spray them out into a vacuum where they dry up and become supercharged so that we can weigh them and analyze them. So it's all super cool science, high-tech, sci-fi kind of stuff. It's a fantastic place to do research and to do great things. So the SILQ Center, what is our mission? We're trying to establish a world-leading center utilizing these advanced high-tech tools of mass spectrometry and other newer methods to discover new proteins that cause neurodegenerative diseases like Alzheimer's and related disorders, and to understand those proteins and what causes them so that we can understand the disease, to develop new biomarkers, to accelerate enrollment in clinical trials, and ultimately to accelerate the development for new treatments for Alzheimer's disease. The center is already launched. We're in phase 1, moving on to phase 2, and there's space being built to make the center even larger, and we're recruiting faculty member and staff to further the accelerated development. What are we after? We're after proteins. I've told you about amyloid beta and its turnover rate. It's incredibly fast. Just in a few hours, you will make a lot of amyloid beta in your brain, and you'll clear it away. And so it's incredibly rapidly turned over protein. ApoE is another protein that we've studied and is really critical because it's the single greatest genetic risk factor for Alzheimer's disease. And I mentioned all the biomarkers on tau, and there's a lot of work that I'm not talking about today on tau and that's underway that we're taking, but it's a bit longer. It's 23 days. But it's not the longest protein. Some of our proteins, like the protein in the lens in the eye, last your whole life. It has a turnover rate of 70 or 80 years, so you won't remake those proteins. Those are set there. Proteins in our skin and other structural parts can be there for many, many decades before they're remade again, and usually only if needed. But these proteins, these neurodegenerative proteins, they are actively made, they're actively cleared away, there's a turnover to them. And that's part of the clue of what we think causes Alzheimer's disease. I've talked to you about a lot of the human studies, but in fact the tools that are at hand in models, cell culture models or animal models, in cells that are transformed. So we can take a piece of skin and transform it into your own little stem cell and make neurons from it, or make fibroblasts, make all kinds of things from it, and then study those cells in a dish. And combining these tools that are present in biology with the advanced tools of measurement and quantitation at the atomic level enables us to discover things that before weren't really possible. And so what do we think is going to be the outcome of the center? What are we really trying to end up with? We're trying to end up with new understanding of the mechanisms of disease, because it's the mechanism of disease is what we develop treatments for. We have to know what's wrong in the disease to develop a treatment for it. And I mentioned developing the new biomarkers and something that's called personalized medicine. It's a popular term these days. 5-10 years ago, it was less commonly used, but what it really means is to identify in an individual, in you, what is special or unique about you, and how does that relate to the disease? Instead of treating everybody all the same, we may have individual treatments, individual tests for specific people. And that's what this kind of thing enables. It enables us to track in an individual person what their current stage is, their current status, and customize medical care for them. So the impact has already been quite large. We've developed the test. Others have been developing tests. It's changing how we're doing things. As soon as these tests came out, the design of clinical trials was changing. Today, trials are launching using blood tests. And so it's already had an enormous impact in the field. And once these things are implemented and when effective treatments are found, recently there was an accelerated approval of a drug in the US for Alzheimer's disease to remove plaques. And it becomes really important then to identify who has plaques and who doesn't. And so these can be used for Alzheimer's disease, but we can translate these over to other diseases like ALS or Parkinson's disease and other diseases. So with that, I'll stop my presentation, and I hope we have a lot of good questions. And I want to recognize and thank the many people that have contributed to this, and especially the research participants. We have people that come in and volunteer to give blood samples and spinal taps and brain scans and do all kinds of work with us to further this research. They are altruistically donating their time. And to those people who've contributed, look at the difference that's been made. To all the researchers, the scientists, the doctors, and everyone that we all work as a team, shown here as my group at Washington University, but really around the world. We collaborate with centers all over the world to make a change and a difference. And these people are extremely committed. Nights and weekends, it doesn't matter. We keep up the fight to keep making progress against Alzheimer's. Thank you.
Thank you. Thank you, Dr. Bateman. We do have a couple of questions from our online guests, and we'll start with those. Question 1, what is the timeline we are looking at for results or impact from your work? This is absolutely incredible.
It depends on what impact is being referred to. So I think one of the shortest timelines I've ever seen from discovery to implementation in medicine is we reported the high accuracy plasma amyloid beta test in the summer, July of 2017. And as of this past year, that test had been translated, commercialized, implemented, and in the clinic. And so that's really quite incredible when I think about it. I mean, 3 years from discovery until implementation and having an impact in the clinic is really quite extraordinary. It's having an impact on clinical trials and research and these kinds of things. So all the tests that are being developed out there, these kind of blood tests are, as I said, are changing what we're doing for research. So it's already accelerating how fast we can launch trials and finish them. And almost without a doubt, that will accelerate the ability to find more effective treatments for Alzheimer's disease. So I think it's already having the impact in the search for treatments. But the question can be brought further. Well, when will there be a nearly perfect test? When will we have tau and amyloid and neurodegeneration and a whole panel? When can we predict when someone is going to get the disease? That's a harder question to answer because discovery is different than engineering. And I was trained as an electrical engineer, so in engineering, you have the answer when you start and you design it to work and you can estimate how long it'll take or what it'll take. In discovery, you don't have the answer when you start. You're looking for an answer. You may or may not find it. It's an uncertainty. And so whether we'll develop tests that can predict how many years someone has before they get symptomatic, we're all working very hard on that, but I don't know if that will be in 1 year or 3 years or 10 years. And in terms of treatments, the development of effective treatments for diseases is perhaps the hardest thing in the world in medicine to predict, or at least that's been my experience. So I don't know, but we already see signs of change in the field along these fronts. So for example, there's several drugs that are in a class that removes these amyloid plaques. They're antibodies, they bind to plaques and remove them. And they're showing that consistently between the different drugs, In early studies, they're not proven, they're not final. We don't know for certain for an individual drug, but they're all pointing the same way, that when we remove plaques, we can slow down the progression of Alzheimer's by some percent. Maybe it's 20% or 25%, somewhere in there. But we don't know that for certain yet. And so when will these drugs be available? I don't know, but it may be relatively soon. But then the next question is, well, what about the next drugs? What's coming next? And there are tau drugs in development. There are drugs that target the immune system in development. And so the field is moving full steam ahead and is growing and gaining momentum rapidly. So I would predict that probably within 5 to 10 years, maybe quite a bit less, we could have a series of drugs that could be used to treat Alzheimer's disease and slow down its progression. We could have several different kinds of drugs that could be used even in combination. And so, for example, one of the things that we're doing in our trials with these families that have mutations is we're bringing 2 different drugs, one that targets amyloid and the other that targets tau, bringing them together in the same patients and administering them, trying to hit both sides of Alzheimer's disease.
Thank you. Our second question is, how was your research impacted by the COVID-19 pandemic?
I think the question is, how has COVID-19 impacted the research? Yes, quite a bit. So I don't think COVID-19 has left anything unscathed. When COVID-19 hit and the shutdowns began, a lot of this research that I described is global in nature. And so we work with partners all over the world. And so when the inability to travel or people to go to work or to do things happens, it ground to a halt a lot of our observational research and even the trials where we were in the middle of trying to administer drugs and treatments to people. Those were greatly disrupted. And so we've recovered from that. We had contingency plans. We've done a lot of work in the background to kind of build up momentum, build up and prepare for the time that people could come back in. And I'm happy to say that we are now operating with people coming back in on a limited and a controlled nature. But without a doubt, COVID-19 had an enormous impact on Alzheimer's disease research, as I imagine it did all research for all medical diseases.
Thank you, Dr. Bateman. The next question is, what is the best way to get into a trial?
There's lots of ways to get into a trial. In the United States, I recommend people do 2 things in parallel. One is you can go directly to the website now. It's called clinicaltrials.gov, and it's all clinical trials that people can enter into in the US are listed there. And so you can search for trials for Alzheimer's disease and they'll show you. It's complex and there's a lot of information, but that is one of the definitive sources. You can also contact a center, a medical center, and ask the Alzheimer's or the dementia group there what trials are available in your area. And so, for example, there are national centers called Alzheimer's Disease Research Centers that are supported and funded by the National Institutes of Health. And these centers are in a lot of the major academic medical centers. And you could contact them and ask, what kinds of trials can I sign up for? I'm delighted to hear the question because there are some real limitations in the fight against Alzheimer's. And despite the fact that there are so many people, more than 5 million people in the United States with the disease, one of the greatest limiting factors that trials have is enrolling patients. And so it really slows down progress when I described how it takes time to enroll patients. So if people are interested, whether you have Alzheimer's disease or don't, there are some trials even before people have Alzheimer's disease, these prevention trials that one can sign up for. So you can go to the National Institute on Aging website, you can go to the clinicaltrials.gov website. The Alzheimer's Association is another great website where they have lots of information about trials and you can contact them. But there's lots of great sources and I do encourage people that if you're interested and you want to help out in the fight, please check into it.
Wonderful, thank you. Our next question is there a correlation between a high IQ and Alzheimer's disease?
There is actually an inverse correlation between education, which is not the same thing as a high IQ, and Alzheimer's disease. So what's been well documented in multiple studies is that people who have lower education, lower levels of education, are at higher risk of getting Alzheimer's disease. And so we think there are environmental factors to this. Now, whether it's the education itself that protects someone, or whether it's the associated factors of getting a high education, that's less certain. But I oftentimes describe the environmental factors in three kinds of categories. One is physical exercise. And so it turns out regular aerobic physical exercise 3 times a week has been clearly demonstrated to protect against cognitive decline and Alzheimer's disease. And there was even a trial that demonstrated that when you intervened and gave people physical exercise, it helps protect and slow down against Alzheimer's disease. So the evidence is fairly strong that physical aerobic exercise is good. It doesn't stop Alzheimer's, it's not going to protect everyone, but it will slow it down and potentially even lower the number of people who end up getting Alzheimer's. And then there are 2 other main categories that the evidence is not quite as strong, but there's lots of evidence like this, evidence on education. And it has to do with diet, so we say heart healthy, brain healthy. So staying away from fried foods and eating more baked foods, avoiding red meats, more vegetables, things like that are good for the brain. And so we want to encourage that kind of good diet. And then the third thing is cognitive and mental stimulation. And we count education as that, but I'm pretty sure dialing into a leading edge medicine presentation also counts. So I think you all have your dose of protection for the day, and you're doing very well. So thanks for dialing in and asking great questions.
Excellent. The next question is, in clinical trials, if a person had a blood test which was positive for AD markers, but they had no symptoms yet, would the person be told of the test results?
In the clinic, we currently do not recommend people who don't have symptoms of Alzheimer's disease be tested with these tests. And the reason that we don't recommend that is because right now we don't have an intervention or a medical management or something that we would do different for that patient. And so if someone is worried that someday they might get Alzheimer's disease but they don't currently have symptoms, we don't recommend a blood test, a spinal tap, a PET scan, any test, right? We recommend doing healthy things, some of the things that I described. We also recommend just seeing if there's any decline. So we can do cognitive tests and follow people over time to see if there's any decline. The other thing, of course, is doing research. Now, I did mention there are prevention trials, and if these prevention trials indicate that doing something before the symptoms begin can be helpful, can help us try to lower the risk of Alzheimer's or treat it before it gets started, then I think tests of people who don't yet have symptoms of Alzheimer's will be, in some cases, appropriate.
Thank you. The next question is, what is science's best guess as to why some people have more aggressive disease, while in others it's very slow?
I don't want to speak on the behalf of all of science, so I'm going to give you my best guess, and then everyone's going to have their own opinion. And the reason I put that qualifier out there is that I don't think we know why some people have a rapid course and other people have a slower course. But it is quite remarkable. So patients with Alzheimer's can be remarkably stable for very long periods of time. I've had some patients 20 years out, and they're just very slowly declining year by year. And other people will have a much more rapid decline over 3 to 5 years. The average duration of the symptomatic phase, as I said, is around 10, 11 years, but there's a huge range in there. And why that huge range exists, I don't think we fully understand. I will tell you that in my clinical experience that People who do regular exercise, aerobic exercise, even with the disease, on average tend to progress less quickly, and it's one of the reasons I always advise people to have an exercise program. The second thing is it could be genetic factors, but there aren't obvious genetic factors that clearly define someone as a fast progressor and low progressor. We do know the Alzheimer's pathology has something to do with it, because the more pathology someone has, the more plaques and tangles someone has, it appears the faster they decline. And so it may be something to do with the amount of pathology. But it's an outstanding question, and one that could be really important, because if we could answer that question, what can really slow down the disease or speed it up, that might be a therapeutic option for how to slow down the disease for everybody or even prevent it.
Thank you, Dr. Bateman. Next question is, like Alzheimer's, has there been work to use biomarkers to test for Parkinson's, much like before the full disease presents itself?
Yes, and there's actually a good amount of work going on in this area. And in Parkinson's disease, there's a different kind of pathology. It's something we call a Lewy body. And these are round circular deposits of a protein called alpha-synuclein. And in that disease, it turns out that there are some assays that have recently been developed that can take that, look for in the biofluid, like cerebrospinal fluid, put it in an assay in the lab, and show that the alpha-synuclein can change alpha-synuclein in the lab and grow and amplify over time and be detected in the lab test. So there's a lot of active research being done in this area now, so that it may be possible one day to even have a biomarker not just track the pathology of Parkinson's disease, the Lewy bodies, but maybe even detect it before it starts, and that then opens up the opportunity for prevention trials.
Thank you. Our last online question is, ancestry tests now include markers for medical conditions. Are these tests accurate?
I can't speak to the accuracy of ancestry tests, because I'm not sure exactly which ones, and I'm not that familiar with it. I'll mention that some of these programs like 23andMe they'll do genetic sequencing. And I don't know what their accuracy is, but I do know that some of them will actually allow someone to see, I mentioned ApoE is the single greatest genetic risk factor of Alzheimer's disease. They'll allow one to see that ApoE genotype that one carries. And you can look that up, and it tells you about your risk, but it doesn't tell you whether you're going to get Alzheimer's disease or not. And so one of the analogies I use for this is, if I told you your risk of falling off the bridge when you drive across it in your car was 10 times higher today or 3 times higher today than it was yesterday, would you go across the bridge? Maybe not. But if I told you your odds went from, say, 1 in a trillion to 1 in 100 billion that the bridge would collapse, then you might drive across the bridge. 1 in 100 billion, well, that's a risk I'm willing to take to get to the other side. And so these risk factors that we talk about, education and diet and exercise, genetic risk factors like ApoE, they change, the numbers sound large. For example, 1 ApoE4 copy will increase the risk by 3-fold. That sounds pretty high. But if you think about it, the risk is not that much higher. It's a few % higher than what it was without that genetic factor at different ages, 60, 65. The risk might go from 2% to 6% for an early onset case. And so, so it's not an absolute. Now, it is cumulative, it does add up, but in fact, there is a risk factor that we can't do much about. It's the single greatest risk factor of Alzheimer's, and it's age. And so as we get older, year by year, our risk of getting Alzheimer's goes up and up and up. And so it may only be a few % when we're 60 to 65. But by the time we're 70, 75, it's 10%, and as we get 85 and older, it can be as high as 25 to 30%. And so if you take the total cumulative, the odds are somewhere in the range of about 1 in 10, 1 in 8 people. If you live long enough, in your 80s, and many of us are expected to live that long, that we have a real risk of Alzheimer's disease. And so I don't want to end the last question on a bad prospect that getting older is going to lead to Alzheimer's disease. It's not the case. Most people actually get older without getting Alzheimer's disease. And so it is not inevitable. There is something we can do about it, and we're all working on it right now. And so there may be a time that we can really, just like we did with infectious disease, bacterial infections, like cardiovascular disease, heart attacks, and now cancer is actually the risk of dying of cancer in some cancers is decreasing quite a bit. And so I predict that that day will come for Alzheimer's disease, where we will be able to treat this disease and move it from a very common disease of aging to a much rarer disease of aging and treat it well. Thank you.
Thank you, Dr. Bateman, for that wonderful presentation. We'll now have closing remarks by our board chair, Dick Miles.
Wow, what an evening. Such an important topic for all of us, directly and indirectly. I'm Dick Miles, the chairman of the board for the Foundation for Barnes-Jewish Hospital. Thank you all for joining us tonight for our final Leading Edge Medicine program of 2021. And Dr. Bateman, thank you for a very insightful look at a topic that affects us all. The last time we saw Dr. Bateman was about 2 years ago at the Foundation when he made a presentation on this. And I was remarking before this started that he's obviously been very busy over this period of time. And then he told me that a lot of what he's talked about tonight was developed in the last 2 years. So this is a rapidly changing field, and it's one that affects us all either directly or indirectly, and one that we are all incredibly interested in. Dr. Bateman's advances in the area of neurology are just a few examples of the impact philanthropy can have. Your gifts move the needle and change the lives of our patients, our societies, and the world. We'd like to ask you to consider sharing something that you've learned tonight with your friends and family. Tell them about our event or share the video of the program. The recording will be posted on the Foundation's website and Facebook page soon. As always, I'd encourage you to visit our website from time to time to learn about the impact that you're making possible. Merely visit us at foundationbarnesjewish.org. Lastly, please be sure to complete the survey about this event to the right of your screen. Your feedback helps us shape future Foundation events. So thank you for your time, and we look forward to seeing you again soon. Good night.